what we are doing there is we're engineering receptor tyrosine kinases. And now, of course, receptor tyrosine kinases play a big role in oncology, including, as we think, in mesothelioma. We have concentrated our work in mesothelioma on fibroblast growth factor receptors, and we do find that they are expressed and that they increase the malignant behavior of mesothelioma cells. So we do think that this is a potential target in mesothelioma. And also there are some clinical studies ongoing targeting the FGF receptor axis. Now, what we have done for this poster is that we created variants of fibroblast growth factor receptors that we can activate with light. This is, belongs to the larger field of optogenetics. So optogenetics in general is an area where researchers try to make cellular signaling molecules controllable by light. This has several advantages. The biggest one is that you can then very precisely, both in a temporal and in a spatial manner, control the activity of these proteins. This has been tremendously influential in the neurosciences already, where people could really, in living animals, actuate signals on seconds and even milliseconds time scales, and then also study by optical means the outcome. Now in cancer research, this is still something relatively new, and we were in fact together with another team in Korea, the first group with our partners at the IST, the first group to generate optically activatable RTKs. Now the idea is that we can use these optically activated receptors for studying the behavior of mesothelioma cells. So we can switch on the light and see with a very high spatial and temporal resolution what the cells are actually doing. So here I think we can get a better understanding of how these signals contribute to malignancy. And the second thing where we think that this might be quite useful is we think that we can use it, and actually have shown that, for drug screening, because normally when you want to stimulate the cells, you have to add a signal, which is, if it's a chemical ligand, you have to add it to the tissue culture. Now light you can simply switch on, and it will in parallel be on in, if you want it, like thousands of 384 well plates. So this, I think, is really a potentially elegant strategy that might also be of interest for pharmaceutical companies. And indeed, we could show with this that we can, for instance, also identify compounds that inhibit ROS1, which is currently an orphan RTK, where the ligand isn't even known. So for these cases, I think that's quite an interesting new strategy.